Welcome to BioInfoBytes. So, you probably know all about the core databases like NCBI and UniProt for sequences and structures, but let's be real. To see the bigger picture, you've got to connect that raw data to biological pathways, to disease genetics, and to all the latest research out there.
So, the big question is, where do you actually find all this crucial information? Well, in today's explainer, we are breaking down 3 absolutely essential databases that every single bioinformatician needs to have in their toolkit: KEGG, OMIM, and PubMed.
All right, first up is KEGG. I want you to think of it as the GPS for navigating the incredibly complex roadmaps that exist inside our cells. Now, KEGG stands for the Kyoto Encyclopedia of Genes and Genomes, which is a mouthful, I know. But what it really is a whole collection of databases all rolled into one. It brings together everything from genomes and chemical substances to drugs and diseases, all to help us understand the high-level functions of a biological system.
In this web portal you're looking at here, this is your gateway to KEGG's most powerful feature, and that's its pathway database. These are these amazing hand-drawn maps of metabolic and signaling pathways. This is absolutely crucial for systems biology because it lets you see how, say, a single drug could cause a whole cascade of effects across the cell, not just at its one intended target.
So how do you actually use it in the real world? Okay, imagine this. You just ran an RNA-seq experiment, and now you have this big list of genes whose expression levels have all changed. You can take that list and upload it directly to KEGG. What it does is it overlays your data right onto its pathway maps, and it highlights which biological processes were most affected. It's not just a list of genes anymore, it's a story.
Okay, so from the cell's general pathways, let's zoom in on human health with our next tool, OMIM, which is pretty much the definitive catalog for genetic disorders.
OMIM, which is short for Online Mendelian Inheritance in Man, is your absolute go-to resource when you found a gene and you need to figure out its clinical significance. It's this huge comprehensive database that documents the relationships between our genes and inherited diseases.
An OMIM entry, like this one for the beta-globin gene, is just packed with information. It's not just some dry description of the gene's function. Oh no, it gives you the entire clinical picture. We're talking associated diseases like sickle cell anemia, and even the specific mode of inheritance. Whether it's autosomal dominant or recessive. It's all there.
And its real power? It really shines in a clinical context. A researcher can just search for one single candidate gene and immediately get this super deep dive into how it's connected to human health. This is exactly how OMIM bridges that critical gap between fundamental molecular biology and actual real-world clinical medicine.
And finally, let's talk about the database that is the foundation for every single project you will ever work on, PubMed, the literature hub.
And this is a fundamental truth in our field. I mean, you can generate the most amazing data, but without the context you get from published research, it's pretty much meaningless. You have to know what's already been discovered to understand what your own findings really mean.
PubMed, which is hosted by our friends at NCBI, is the world's biggest and most important repository of biomedical literature. It's a totally free service that gives you access to citations and abstracts from Medline and a ton of other life science journals.
And the scale here is just staggering. We're talking about more than 35 million citations. This is it. This is the definitive archive of our collective biomedical knowledge. If it's been published in the life sciences, you're going to find a record of it right here. But listen, here's a pro tip. Don't just use simple keyword searches. To really unlock the power of PubMed, you need to be using the advanced search. You've got to use Boolean operators, and, or, and not, and apply filters for things like dates, article types, or even species to really zero in on the exact papers you need.
So how do these 3 tools fit together? They aren't just 3 separate websites you visit now and then. You should think of them as a complete, cohesive toolkit for doing advanced bioinformatics research.
The key takeaway here is actually really simple. For understanding biological processes, you go to KEGG. For investigating genetic disorders, use OMIM. And to put it onto context, you've got to consult PubMed for the literature. And that is your bite-sized breakdown. Mastering these tools isn't just about finding data. It's about being able to ask better, smarter questions. What's the story hidden in your gene list? Next time, we're diving into essential biological file formats. Make sure you subscribe, and we'll see you on BioInfoBytes.